So welcome. We're going to try and get going this morning. Welcome to day 2 of our conference on reimagining human health, the microbiome, farming, and medicine. Yesterday there was a bit of, the way we structured the conference is after an introductory panel on the microbiome, we looked at a couple of different areas: soil and water health, farming with both animals and plants, and then medicine. And then we had a great final panel with student questions. Today, we're going to start again with a general panel with a different kind of introduction to the microbiome. So Larry Weiss, on my right, has been for a number of years working with, he'll tell you about it, but with the tribe in Brazil. Tribe that Venezuela, in the Amazon. I guess it goes from Brazil to Venezuela. But it's a tribe, the Yanomami, which I assume, have any anthropology students here at Bard studied the Yanomami? They're probably one of the most interesting anthropological tribes because they're the tribe with the least Western contact. We're welcome to sit down if you want. It's up to you. I don't mean to make you stand. That we have. And so it's a tribe that I've read a lot about, and when he told me about it, I was like extremely interested in it. So I'll let him tell you about it, but it's about how he's gotten access to, in a sense, their microbiome, and it's a way of comparing a largely undepleted microbiome with ours and what we can learn from that. We'll have commenting on it Rodney Dietert and Laura Kahn, both of whom spoke yesterday from different perspectives, and we'll bring in your questions. So I'm excited to get going, and I don't want to spend too much time because I want to have time for discussion. So, Larry.
Roger, thanks for inviting me, and thank all of you attending, particularly those of you with your entire careers in front of you. I won't call you young people, although you are. Oh, I just did. Okay. What I'm going to do this morning is I'm going to take you on a journey. And we're going to start, because we're going to go back in time. But the ideas I'm going to talk about evolved in the minds of men, so we have to see how we got here. So I'm going to start by talking about how it is we got here with the microbiome. Then I'm going to talk about a little bit of what we've learned so far, and how what we've learned is transforming our notion of what we mean by health. And I hope to inspire you to participate actively in that transformation. Then I'm going to talk a little bit about what we have learned by working with these indigenous people. It's like setting the clock back 12,000 years to see what our microbiome looked like before our biology and our behavior diverged. And then at the very end, I'm going to challenge your notion about what it means to be human and the world in which we live. This translates basically to say the end depends on the beginning. Your first step into an unknown territory determines what each other step will be. So we need to go back to the beginning a little bit. And this was a statement by Francis Crick of Watson-Crick fame, where this is where the microbiome started. And in a little remark in a paper that was about decoding hemoglobin, he suggested that by looking at these sequences, we could develop a taxonomy or a biological clock to determine how life evolved. This was picked up by Linus Pauling. This was in 1958. This was picked up by Linus Pauling and Emil Zuckerkandl, and then picked up by this man, Carl Woese. Carl Woese was not a microbiologist. He was a molecular biologist, and he came into this field with the notion that if we just followed proteins, we could only go back a couple of 100 million years. His idea was, his inspiration was, let's look at the thing that makes proteins, which is the ribosome, and he started sequencing this when it was very laborious, and he came up with a new taxonomy. I've got a thing here, okay, which is down here. And like any revolutionary, and that's what he was, he came up with a new kingdom, which is archaea. So it wasn't just bacteria and archaea. And this did not go well for him. When you come into a field with new ideas that challenge the dogma, don't expect it to be easy. And he suffered for this. But it illustrates a point, which is the person who's going to change your field tomorrow probably doesn't work in your field today. So then we got interested in who we were. We decided we're going to sequence our genome to discover what it meant to be human. This was a huge project that started in 1984 and finished in 2003. We developed the technology to do this, to advance the rate at which we could read our DNA. And that tool When we went looking for our humanity, we discovered that we were animals living in an almost exclusively an ancient microbial world. We had a microbiome. Before, microbiology was purely descriptive. If I couldn't culture it and see it and measure some of the way its chemistry worked, it didn't exist. Suddenly, this blew things up, and the technology got better and better, and we could look deeper and deeper. This is the Human Microbiome Project, which led to the second Human Microbiome Project. And suddenly, we were overwhelmed with all this information. We got very excited. We knew a little bit, and we thought we knew a lot. And suddenly, there's all these companies that come in, like neo-colonialists, on a new technology to figure out, how do we make a whole bunch of money from this? And it's still really early. I want you to remember that. This is where the first step in a transformational science. And this is what's known as the Gartner hype cycle. And this is how this always works. We get brand new technology, and this is going to solve anything, and we're on this peak of inflated expectations. It's going to solve all our problems. Everything's going to be better. And then we discover it's actually harder because we only knew a little bit. And we go through the trough of disillusionment, and those of us who keep on persisting and working at this hopefully get us to a plateau of productivity where we come to understand a sense of wonder and humility. Science is the disciplined practice of wonder. So what have we learned so far? The first thing we've learned is it's still early. We only know a tiny little bit. The foundational science, our ability to sequence these things reliably, even within a given laboratory, has bias. So from one day to the next, from one operator to the next, we're still getting different answers. Go to different laboratories, and you'll always get different answers. We'll get there. For those of you who remember when your phone first had a camera on it, we're there. You could tell they were people. You couldn't tell who they are. And we're going to keep on getting better. But what we've learned, it's incredibly complex. When we get better, it's not going to get easier. So I'm going to give you an example of this. This is the NASA Ultra Deep Field experiment. What they did is they pointed the Hubble at the darkest piece of space they could find, and they had it take a picture every time it went around for about 400 days. With 21 days of these images, the darkest piece of space showed them 10,000 galaxies. And if that's amazing to you, imagine that wallpapers the entire sky. And at that end, which is the far end of large things, that's a big number. You have more bacteria on you than there are galaxies, even with that. So it's going to get more complicated when we get more data. Grow a sense of humility for science. This is where we are. And by the way, I exaggerated those two slices of the pie up there because otherwise you wouldn't be able to see them. We don't get certainty in science. People believe that as scientists, the substance of what we work on is facts and truth, but in fact, that's not correct. What we do is we gather evidence, we build models, we ask questions, we get more evidence, and we keep on repeating this. And the best we can hope for is what we know today will be a gross oversimplification of something so complex that we can't yet even conceive of it. And much of it will be wrong. And that's fine, because being wrong is how we get to being right. It's a lifelong pursuit. Okay, this is one of the things we've also learned, and I'll get to this when we talk about the Yanomami in a moment, is we've lost a lot of diversity. We used to have lots of bacteria that we no longer have anymore. And it's not just the taxonomic diversity, which is who's there, but it's the genetic and metabolic contribution they make to our physiology. We lost not just their presence, but we lost their contribution. And the hardest thing to find is things that you did not know you lost. And if none of us have them anymore, where is the reference point? Where is true north? Second thing is I want to advance. We've all heard of a germ theory of disease where the presence of some bad actor causes illness. I want to advance a germ theory of health where it's the absence or an imbalance within this ecosystem which predisposes our physiology to illness, and we only see the very last step. We don't see all the preceding steps that led up to this. We're not at war with the microbial world. We emerged from it. It nurtured us, and we're immersed in it, and we're only beginning to appreciate it. It's only about 20 years we even started to admit that we had a microbiome. And it may be that our microbiome has us, by the way. So here's the big question for all of us to ask today, which is, are we going to treat disease? Are we going to keep fixing broken stuff? Or are we going to start asking, why is there so much broken stuff? Evolution does not optimize organisms that have so much broken stuff. So what happened? Is there a way to restore health? And is that an interesting problem to spend your careers working on? And I suggest that it is. But if we're going to do that, we have to answer the question, what is health? So I did some research, which is to say I Googled it. And what I discovered is health, almost all of us define health as not being sick, that I know of, yet. That's why I don't go to the doctor. He might tell me something and discover I'm sick. Okay, but that led to wellness, so I decided to Google that, but that wasn't particularly helpful because that led back to health. And the FDA doesn't have a point of view on this because that's not what they do. They are a disease organization, not a health organization. And this is where we are today in the United States. This is life expectancy as a function of health expenditure. And this is all the other developed countries in the world, and they live longer and pay less for it. We pay more and live shorter, sicker lives than everybody else because we optimize for incomes. We practice profit-based medicine. We don't practice outcome-based medicine. We made incomes more important than outcomes, and we got exactly what we asked for. This is an exhibit at the British Museum. It's called Pills of a Lifetime. These are the pills you'll take in a lifetime. Now, it's not as bad as it looks because one side's a man and one side's a woman. It's 16,000 pills, in case you're wondering. But it's actually worse than it looks because this is just prescription medications. Throw in all the other stuff, and it's 3 times this size. That's what we call health today. But I'm going to argue that health is more than just not being sick. Health is an evolutionary legacy. Biology gave this to us, and we've lost it. And here's how I got here. I'm going to introduce a word to you. This terroir. It's from French viniculture when they grow wine, and we always thought of it as the climate and the soil's impact on the plants that grow there. But it is literally the biological impact of all of the microbiota in the environment and all of their genetic and metabolic contribution. We can't take ourselves out of our terroir. We live in our terroir, whether it's in this room or in the jungle. So some work I did with a previous company led to the Yanomami, and this is some work that was done by Marty Blazer and Maria Gloria Dominguez-Bello, his wife, and they went and lived with this group of Yanomami, and they reported that they had the highest diversity of bacteria and genetics functions ever reported in a human group. Also, they have antibiotic-resistant genes, the antibiotics they've never been exposed to. Now, if that sounds surprising to you, it shouldn't, because our antibiotics all are directly or indirectly derived from bacteria and fungus themselves. In fact, we think of them as antibiotics, but these are probably the important communication molecules that these ecosystems use to maintain their balance. We took them out of context. I'm going to come back to that idea again. And what we did is we turned them up to 11, to take a line from Spinal Tap. And what happens is we then activate those genes. We turn them from being just in their DNA. It goes into their RNA because we activate the machinery. And you can end up with horizontal gene transfer into organisms that never had them. I'm getting there. Okay. Yeah, but if you look at their skin, what you discover, and you can do this yourself, you can Google Yanomami and image it, they don't have any of the inflammatory diseases that we have today. Acne, eczema, rosacea, psoriasis don't exist at all. And it's not just them. Other groups of hunter-gatherers have been studied, whether in New Guinea or in Paraguay, and they also don't have any inflammatory disease. And this is kind of interesting. This is what we look like today. I know that's gross there. That's gone. This is a group that went and looked at, they did non-contrast coronary artery CT on 705 hunter-gatherers in Bolivia. I have no idea how you do a thing like that, but what they discovered is they don't have coronary artery disease. They live a largely traditional lifestyle. They are impacted, you can tell from the clothing and things, but they live basically the way they've lived for the past 11, 12,000 years. And this is their data. That's us on the left. This is coronary artery calcification. You want to look on the right, but on the left, that's who we are. They don't have coronary artery disease, they don't have diabetes, they don't have metabolic syndrome, they don't have any degree of inflammation. But if you look at their C-reactive protein, which we use as a diagnostic test for inflammation, half of them would qualify as being inflamed by today's criteria. which tells me we don't know what a healthy human looks like because none of us are healthy. We're all missing pieces. And this is another graph on this. This is them down here at the bottom. That's the rest of us. If you had a choice, you'd probably be a Japanese man. And this is, I think, how we got here. We were optimized by biology. We lived in our terroir for long periods of time. We had a relationship with our microbiome, but we were hunter-gatherers, and hunting was work, and so was gathering. So we developed agriculture, we domesticated animals, we started living in cities, then recently chemistry, technology, and consumerism, and we broke connections with our microbial world, which predisposed us to these inflammatory diseases. And I don't know if these numbers are correct, but this is what the perceived message out there is that we are sick. We are living in an age of epidemic inflammatory disease. And it's not just me who feels this way. This is Risa Levizo-Amore from the Robert Wood Johnson Foundation. We're raising this first generation of children who live shorter, sicker lives than their parents, and that should concern us. Why are we so inflamed? Maybe this is our biology reaching out and tapping us on the shoulder, trying to get our attention while it still can to remind us who we are and what our place in biology is before it's too late. So, this is David Good. David is half Yanomami. The question to ask is, can we live in a modern world without going back to live as hunter-gatherers with some of the health the hunter-gatherers have? So David approached me. David's mother, Yarima, is full Yanomami. She lives deep in the jungles in Venezuela. David's father was an anthropologist. He went and lived with them for 12 years to study them. During his tenure there, the head man of the village betrothed his sister to him, as one does, and he married her and had 3 children. David is the oldest. She lived in this country, in New Jersey, for 5 years, doesn't like the way we live. They took her back. David didn't see his mother for 19 years, got a degree in biology, mounted an expedition, and found her. He'd been there, which is astonishing because they're semi-nomadic, and then he'd been back there 12 times, and then he approached me to work together to study their microbiome. This is how you get to where his mother lives. This is insanely difficult. First of all, it's Venezuela, which gets extra difficulty points right now. When you fly on these little planes, and then you get to this place and you go up a rapids in a little canoe to get to this village that's called a Shibona, where they live, and he goes there to see his family. And this is his family. And this is dinner. They live on things like boa constrictors and plantains and tarantulas, and this is a peccary, by the way. Interestingly, whoever brings in the game eats last. These people live these are the only humans who actually know how to live sustainably within their environment. And this is David studying his family. I want to point out there's a little blue stick. These are called hee-hee sticks. They use these for endorment. That's one of the sticks he uses from doing the swabs. They have a tradition of generosity. He was supposed to bring those back, but he couldn't deny it to her when his, I think she's his niece, asked for it. So this is the data that we found, and I want to talk about this. I'll give you a little bit of background on how we do this. You do these swabs, and you're looking for taxonomic diversity, which is how many different species are there. And down here on the left, the red group, that's us. That's a group from the Human Microbiome Project. This was swabs from the upper back. And that's, on left, it's called chow diversity. That's basically the number of different types of bacteria. It's almost strictly a number thing. On the right is his family. And the first point here is David before he left the US. And you can see as soon as he hits his mother's village, right in the middle, he comes up and he starts looking very much like them. Suddenly, you put him back in his terroir, you put him back where his family is, and he acquires all this diversity that he had lost. These are pie charts of the bacteria that are on him. On the left is basically what we look like, and what you see is a couple of really huge pieces of pie. That's what we look like. This is at his maximum diversity in a village. Suddenly there's a lot of really small pieces of pie, and on the right is his mother. So he started off looking like us, and within a week of being in their village, he starts looking like them. We can go back. You can put them back. And this is just another way of looking at it. This is looking at gene pathways. This is called gene ontology. This is from looking at the RNA. The DNA is the blueprint. The RNA are the work orders. And he started over there with a couple of really big things and not that much of this other diversity. And he starts moving and looking very much like his mother did. And the question is, what genetic and metabolic machinery has he acquired, and what is the implication of health? And I put this up there just to show that David, you see here, this is the biologic processes. The left, where it says healthy skin, by the way, it's mislabeled, we're not healthy, that's us. The blue one, the blue circle, is his family. And in between, you can see that David lives between two worlds. Same thing with his molecular functions over there. I'm going to ignore this one over here, but this is antibiotic resistance, and you see that basically them, us, and him all overlap. We all have the genes for antibiotic resistance. These are not things that we acquired through mutations. It's other bacteria in them that don't have them that we can trigger their acquisition through the inappropriate use of antibiotics. It's the movement of those genes into places where they didn't have. It's not the presence of the genes in the first place. They are everywhere. There's a recent paper that found them deep in the caves in New Mexico, where it's been isolated for at least 2 million years. So I'm finishing up here. Our health and our fitness is not a property of us. It's a property of the entire ecosystem. And the ecosystem is the entire planet. There is no other. We are animals immersed in an ancient, almost entirely microbial world. And to illustrate this, this is the most recent tree of life based on our sequencing data. Everything you're seeing here is microbial, and this little green branch over here, that's the eukaryotes, those of us who have those species that have nuclei. And down here, the Epistoconta, this tiny little branch, that's all animals and fungus. We share a tiny little branch. We're newcomers. We live in a microbial world, just because we couldn't see it. And this information is ancient, and it is what keeps us alive. And we're in the midst of a scientific revolution. We knew a tiny little bit before. We were able to do pharmacology. We could draw little diagrams, A plus B goes to C. We were Newtonians. We're now moving into this much more complicated systems biology world. Today, in the life sciences, we are where physics was 100 years ago. And you can imagine these Newtonians watching this oncoming chaos, denying that it's going to make a difference. They weren't wrong about what they were doing. It was just a small piece of a much larger, much more dimensional world that they couldn't imagine. Imagine a color you've never seen before until someone shows it to you, and then you can never go back to where you never saw it. And this is where we are today. This is the world that you will have careers in if you choose this science. It is an incredibly exciting time to do it, but there's also a responsibility that we do it right. And I'm going to finish up with this. Our perspective, how we see the world, how we see ourselves, is constructed of how our identity and the context of the world, and that creates this perspective. And this idea of who we are was created in the mind of Descartes in 1643 when he told us, we think, therefore we are, and that we were separate from everything. And I'm going to illustrate you through, I'm going to give you a word, and I want you to call forth an image in your head and don't tell me what it is. Ready? Here's your word. Everybody got an image? How many of you had any people in that image? Raise your hands. 2. I've been doing this for a long time. Usually it's 0. We took ourselves completely out of context. We saw ourselves as a noun. We're a human being separate from all other things. Descartes told us the rules didn't apply to us. The rules do apply to us. We're not a noun, we're a verb. We're being human. We're a complex process of processes existing in a much larger, much more dimensional process of processes, and energy and matter and information flow through this all the time. And when we take ourselves out of context, we are suffering from an existential identity crisis. All of the problems that we have described here and yesterday are because we think the rules don't apply to us. And they do. And when biology comes back and says, By giving us all this information, we're getting something wrong. If we don't pay attention to it, if we don't start working on this problem, we will pay the consequences. This is a very exciting time to be going into this field. Let's take it seriously. I'll see you down the trail. Thank you.
So one of the things I love about hearing Larry talk about this, besides his excitement for it, which is really catching, is that he brings it up to this question of what are we as humans. And I talked a little bit about that yesterday at the beginning. But Hannah Arendt, when she writes a book, The Human Condition, is basically saying that we are conditioned animals. We are not. There's not some abstract human nature, and that we live, and part of being human is to be in a world that we don't control, that we're born into, that we can't. And what makes humans different from all other animals is we build the world. We make an artificial existence, and then we live in that, and that artificial existence then conditions us. And what she, in that book, The Human Condition, says is we may eventually live in a world that is fully artificial. We may live in a world like a spaceship, where even not every environment, every water, every drop of water is made by humans, not natural. And even every human is in the end made by humans. And what would that mean for the human condition, to lose chance, to lose that? And so these are the kind of questions she did ask. And she said, in the end, my job is not to judge them per se, although I have my judgments. It's to help you think what we are doing. And in many ways, that's what this conference is about, is to push you all to think about what we're doing. And one thing that, if there's one takeaway, I think, from this day and a half, and hopefully 2 days, is we are destroying the microbiome. We are destroying this place in which we lived. This symbiotic relationship between the humans and whatever we call them, fungi, microbes, biomes. I never actually know what the word is.
So that's a controversy, whether or not it's microbiota, which is, in one concept, is all the organisms, and the microbiome was the genome. But the current use of the language is, when we refer to the microbiome, we're referring to all the organisms. That seems to be where the language is going. But just recognize there'll be people who have different opinions on this that are based on their understanding of the language. And let's try to reach a consensus so we can communicate effectively.
Great. So, this is, I think, somewhat of an existential question. I mean, I'm sure many of you, a lot of young people here in the audience, shout out to the Bard early colleges from New York who are here. Today is a strike for a global climate change strike, and I appreciate you guys being here and not at the strike. Come and learn something. In many people's view, and I'd like to hear all of your views on this, A, this is connected to climate change. And B, a couple of people said they think this is a more dangerous issue for us, or at least a more timely issue than climate change. I don't know if everyone agrees with that. It's a question I have. I'll put it again to people in the final panel today. Anyway, I have two real experts here on the microbiome, which I am not. And I thought I would let them comment on briefly and question, and maybe pose one or two questions to Larry and get a conversation going. So we'll start with you, Rodney.
Okay. Oh, Larry, brilliant presentation. Really important. Thank you so much. Couple perspectives. This is an opportunity to examine one particular group of people. And we recognize that there were originally other indigenous people that had not been affected also by what we would say is modern life. And they lived in other areas and had other foodstuffs, and the geography and their climate and their foodstuffs and soil microbes may have led to a somewhat different mix of microbes, but nevertheless the diversity issue, I mean, from what people have seen that have studied other people more impacted, but still living more in their historic ancestral lifestyle, that they have more diversity. So that kind of holds. But the idea of the perfect microbiome, I think people now think that there are several different healthy microbiomes depending on where you live. And I know Singapore researchers have presented that they can analyze the microbiome in a family and tell whether you're eating Japanese rice Chinese rice or Indonesian rice, that they have fingerprints. So keep in mind that ancestral diet and environment, the whole milieu, will determine your particular mix. But this idea of diversity and genetic and metabolic diversity and function is critical. And this is the pinnacle of our example, and so important in, I can't tell you how important Larry's work is, in our potential to restore what we really are. I wanted to just comment on a couple of areas, because we talk so much about health, and I know in my book I give an example of airplane seat selection to actually, and I won't go through the exercise, but to argue that it is very difficult for us to congregate and socialize like our ancestors used to. Food allergies for a child's birthday party. Again, the airplane thing is, you have, Mayo Clinic says eat peanuts if you have diabetes, but you may have a peanut allergic person next to us. We have service dogs and we have dog allergic people. So there's a lot of reasons why your plane might not go. It's hard to get people in the same room, and that's untenable from what our ancestors used to do, from what the Yamanaki do. So there is this socialization that I think we actually are losing. And we're losing it from sort of outpicturings of our condition. I'm very interested in to me, it seems like we're also becoming dull. And I mean dull, not as interesting people. We're very interesting people, but our senses. So I'm very interested. The microbes have interesting roles in our physiology, including, you brought up wine, and that's actually what triggered this. Because if you're a great wine connoisseur, if you can detect a lot of different subtleties in wine, I'm not, but I'm a toxicologist, so I didn't decide. If I didn't drink early, I'm not drinking late in life. But the flavor detections, the zest of life, that's thanks to your microbes. If you've got that palate, it's not so much your mammalian part, it's your microbes, because they are reacting with the grapes. I mean, the grapes have their own microbiome too, but your palate, your oral microbiome, is giving you the senses. Well, the nasal microbiome and odor detection. Every way we sense our external world is driven in part by our microbes. And losing that diversity makes us dull in senses and makes us not really have the experience with nature, the experience of life that to me is core. I want us to recapture that along with other ways you measure health. We need to be bright, curious, and embedded and enjoying all that nature offers, all that we are a part of. So I just make that comment. I don't know if you have any.
Yeah, two things. One is the consistency of their terroir from the time they're born until they grow old is very different than us. So it's not just who's there, but they have accommodated to them from their very birth. And there's a great deal of difference between what they have and what we have. So as one example, on their skin, we've already found they have bacteria that make UVA and UVB absorbers. They have bacteria. Which means what, Larry? They make sunscreen. They have bacteria on their skin that protects them from sunscreen. They have bacteria on their skin that makes vitamin K2. They have bacteria on their skin that make retinoic acid derivatives, which are anti-inflammatory. In their gut, we all regard bifidobacteria as part of a healthy gut. They don't have bifidobacteria. I suspect, and this is a complete conjecture, supported by 2 data points, so it's just the start of a conversation. I suspect we picked up bifidobacteria when we domesticated animals, and we started living with them. This is just a data point. There are other in the Amazon, there are other uncontacted people besides the Yanomami. There are probably 20 or 25,000 of these humans alive today. Nobody thinks they are going to survive. The Yanomami are interesting because they don't have a written language. They don't count time the way that we do. In fact, they don't count past 2. It's just 1, 2, and many. The entirety of their incredibly rich culture is carried in the minds of their elders. And every time an elder dies, it's as though a library was burning down. Unfortunately, the land upon which they live has gold and oil under it, and it's in Venezuela or in Brazil, where they're unstable politically, and they seem to think the way to protect this asset is to capitalize it. We have been told that we are the pinnacle of evolution, made in God's image, crown of creation, what have you. We behave like spoiled, entitled beneficiaries of a 4-billion-year-old biologic trust fund upon which we depend for our very survival, and we seem determined to squander. We need to protect these people because they are the last remnant of what healthy humans look like from an evolutionary perspective, and the way we protect them is we leave them alone.
We're gonna go a little bit late because we started late, but let's try and keep it brief and make a comment or question. Great.
Yeah, so, well, this was a fabulous presentation, thank you. And it makes me think, well, we are obviously not going to all return to the type of lifestyle or the terroir or the environment in which the Yanomami live. So it makes me think that maybe 10,000 years ago when we domesticated plants and animals for agriculture, it was a double-edged sword because we've changed basically our whole way of life from being hunter-gatherer to now being agriculturalists. And while it's allowed us to have civilization, we have paid a substantial price, not only in zoonotic diseases, but in the changing of our entire planet and how we use its land, and the buildup of all of our wastes, whether it's contaminating the soil, the water, or the air. We are contaminating our microbial world in ways that we don't fully understand, and we're getting sick. Relying on antibiotics to try and kill off the bugs that are making us sick, but we have to start thinking of antibiotics kind of like pesticides. They're killing off the good bacteria and the bad bacteria, and it's not really in our best interest to rely on them. It's not fully sustainable, and we really need to start working with nature, not against it, which is what we've been doing now for quite a long time. So it also makes me think, how are we going to practice medicine in the future? How are these discoveries going to impact your physical exam, your checkup, if you will? Medicine is very conservative. It doesn't like to change, but I think perhaps this will have a huge impact, revolutionize medicine in how we define who's healthy and who's not. So I'm curious, Larry, if you have any thoughts on that.
Maybe you can, I mean, I know you're thinking about that question of how we practice medicine and how we practice health, it maybe is the way you want to put it. Could you just give a quick response to that?
Sure. Quickly, medicine today is about fixing broken stuff, and we're constantly fixing the last thing that we broke, but the consequences, which we call side effects, are higher order failure modes. If we don't know what health looked like, so restoring health is not preventing disease. Restoring health is trying to put back things that we're missing which predispose us to disease. Those are two different activities. So the answer to what medicine will look like in the future, I hope someone in this room will show us. And it's beyond my power of, I don't have that kind of insight. What I think we are going to learn here is that once we develop some humility, and recognize that we really don't know very much, we will be able to approach this with a beginner's mind. The beginner sees lots of possibilities, whereas the expert only sees a few. Carl Woese came in from the outside. Martha Carlin, who's the reason I'm here today, did not train in the sciences, and yet she is more qualified than most people who work in the microbiome to opine on what the future might look like for this. So it's a great question. Please answer it for us.
So we're going to start taking questions. I might take two at a time so that we get a number of ideas in the room. Are there questions? I see one over here for that mic and this mic right there to Isabel. We'll start with two questions and then we'll let you sort of, anyone want to speak to them? Stand up.
Oh, okay. Oops, yikes. Hi. My question is about ethics. And I think that we should spend some time thinking through the ethics of this kind of research. Because even in the language that we use when we talk about the Yanomami as them and what they have versus us and what we have, you said something very interesting. We should leave them alone. Doing this kind of research is, in a sense, unethical, right? Because of the very nature of how we share and exchange microbes. And even the ways in which David's his name?
David.
Right? David himself is steeped in a web of like historical ethical quandaries because his father was a graduate student of Napoleon Chagnon, who described the Yanomami as the most violent people, fierce people on Earth, right? So there are ethical implications to this kind of research, and I wonder how we actually limit or even stop, maybe, individuals who could look at the Yanomami and say, or even other indigenous or Amerindian individuals and say, well, if we know, for example, that there are these groups of people who have these particular markers, and if we actually take seriously the claim that this could be a bigger crisis than climate change, how do we then really navigate staying away and leaving them alone when right there could be the answer to all of the questions we've been asking over the last 2 days?
Great, thank you. Do you want to answer?
I do want to answer this,
Okay.
because that is a really important question, and I think about this a lot. So we have no choice about who our parents are, so David was kind of stuck with that. And he didn't see, and he's written a book. I urge you all to read it. It's called The Way Around. You can get it on Amazon. David came to me. So we have no right to show up at any of these indigenous people with an IRB or an informed consent, because those concepts have no meaning to them, and then get a thumbprint on a piece of paper and start collecting samples for them or doing anything. For these people, land rights are human rights. And so we need to leave them alone. What happened in this case, and that's what makes this so unique, is David came to me. Again, he trained in biology. And he came to me because he was concerned about the survival of his family. And that's really what was at risk. Because, they said, their land has gold on it. And the gold miners come in and they bring diseases. When you do that search for Yanomami images, you'll find some images of Yanomami who get measles and they're dying. And they're dying of malaria and they're dying of TB. So the way we've addressed this is the following. One of the beauties about the type of work we're doing is once we have the biobank, and we're building a biobank of these ancestral bacteria, we never have to go near them again. But they own the biobank. David and his family have a not-for-profit, and they own the biobank. And to the extent that there are proceeds or profits from the development of this biobank, they participated in the following way. One, the Yanomami who no longer live a traditional lifestyle, these people are dying, they don't have health care, and they're dying of malaria and measles and tuberculosis and other stuff. We'll get them health care. We get them anti-malarials because they need them, because they're dying. And then we train them to be rangers, to protect their indigenous and minimally impacted and uncontacted brethren. It's the best we can do. But I open this conversation up to all of you because we really struggle with those ethical questions. And by the way, there is no black and white. It's everything is gray here. They are entitled to survival, self-determination, and agency. But right now they have none of those things. And so this is David's effort. This is a young man's effort to save his family's existence. It applies to their village. Their village is about 25 people today, at least the last time he was there. That's what it was. And they're all related by blood or marriage. Now, one other wrinkle to this, because I know I'm supposed to be brief and I'm not good at that, is that these are indigenous people. If this was a family of white people living on a ranch in South America, and they had something of value on that ranch, and they wanted to start a business, or they wanted to do something with this, no one would blink an eye. Anytime you do anything with indigenous people, you have to be especially careful. But we didn't reach out to them. He came to me and said, help me with this. And he'd been trying to raise money. He'd been running around giving talks, heartbreaking talks, by the way, and was able to raise small amounts of money. I'm a venture-backed startup doing basic research with indigenous people in the Amazon. And if that is the most improbable sentence you have ever heard anybody say.
Thank you. Isabel, I know you had a question.
Thank you for the question, by the way.
Hi, you ended your presentation on a really hopeful note, and you said we can restore. And you just mentioned that there is a genome bank or a biobank. So does this mean that the large majority of people who are sick, because that's the implication, that most people are sick, does that mean that we can, those people who have ability to get to a biobank have the ability to restore themselves, and everybody else is, what's the nice way to say it, not in front of kids? Effed, screwed, it's not going to be successful for everybody else. And so is the hopeful part, was that just to make people feel good, or is there really a happy ending here, and how does one go about that?
Again, great questions, thank you. The idea that you can restore bacteria, and if you're not within the terroir where those bacteria exist and have them stay, this is the whole issue of colonization resistance, and I think that that's a pretty steep hill to climb. But the question of whether we can restore the missing genetic and metabolic function, I think that there are strategies by which we can do this. By the way, I didn't say that some of us were sick, I said all of us are sick. Just to correct that. But what makes me hopeful about this is that I believe that once we know what health looked like, once we have a sense of true north, and not just from them, there are other ways of trying to plumb this, but we have to start asking the question. Everything that we do in healthcare and medicine is about treating disease and fixing broken stuff. You make lots of money doing that. It's great business. I think Novartis just released a new cancer treatment for $2.5 million a pop. Everyone's making money and we're getting sicker. I want to inspire us to start asking, what does healthy look like? And how do we get there? How do we get that journey? So it's bigger than just restoring the missing genetic and metabolic function. It's a change of mindset.
Larry, can you give us an example of what it would mean to use what's in the biobank as a way of restoring health? I mean, just one example that would be, that's concrete, that we can latch onto.
So I'll give you an example, though it's what I did with AO Biome and with Mother Dirt, which was the consumer side of this. These were ammonia oxidizing bacteria. They're ancient autotrophic bacteria. That means they cannot use carbon for a fuel source. They burn ammonia, and they convert it to nitrite and nitric oxide, and they're found everywhere in nature, but they're not on you anymore, because soap and surfactants and preservatives and everything that we do kill them. But it's important for our health. Not so important that if you don't have it, you die, but important enough that it helps maintain your skin and your physiologic health. And so what we did is we put them into a bottle. Live bacteria into a mist. You can buy this if you like, and spray it on your skin. And we found a number of interesting things, but one of which we found in a safety study is spraying these live bacteria on your face twice a day reduced your blood pressure. Okay, if you keep your eyes open and you maintain a beginner's mind, you see stuff you never expected. So what will it look like? I don't know, maybe something like that. Maybe something that none of us have thought of yet. We don't know until we look. If we knew what we're doing, we wouldn't call it research.
I'd love to get some questions from the students in the audience, if anyone has them. So think, and I'll start calling on you if you don't raise your hand. We're gonna have a question up here, and then this young man there, those two next. Let's take two questions if we don't mind, because we're yeah.
I've been a lifetime learner. So
Linda, there's a mic.
I think I'm a student because I've been a lifetime learner. So our microbiome has some things in it that under different circumstances can make us sick as well as be part of our microbiome. Did you find some of those things in the Yanomami? Like somebody yesterday said, babies have C. diff. Do the Yanomami babies have C. diff?
No, we didn't.
We're going to take two questions. Is that all right?
Yep.
And then you.
Hello. Okay. Oh, it's close. I heard you earlier mentioning how we've become quite dull, as you had said, with not specifically like personality-wise, but but you had said that we had specific, like certain microbes that we no longer had that could have like helped us, you know, engage in different experiences that we can no longer have access to. And when you say we, who exactly do you mean? Do you mean Western people, or are you referring to us as a global community? When you said we had lost the taste to taste different types of wine.
Great question. So two great questions.
I'll take yours. You take okay. So the answer when we develop a bad relationship with something within our microbiome and we think of it as a pathogen, that may have precedents that we have not seen. But the answer question about C. difficile, no, we didn't see that there. We saw some things there that have never been seen before, and we found, by the way, on the skin, over 800 metabolites that have never been reported on skin before. It's still so early. I mean, so even if they had it there, and we didn't see it. And by the way, we looked at the, for the sake of the younger, we looked at the poop of a 1.5-year-old who is still breastfeeding. And typically, those would have lots of Bifidobacterium longum infantis if we followed our current, and they didn't have any. They had chloroflexi, which you may know what that is, but I didn't either, and I had to go look it up and didn't find very much. So this is a crack in the door of an entire new universe of what it means to be human.
Yes, thanks. Great question. And I think when I'm referring to, well, first of all, there are gradations. So when I'm referring to we, I'm referring to those of us that have been most immersed in probably the U.S. westernized food megacities and the like. So, you know, we moved off the land, we moved away, you know, we don't visit farm animals that often, and as a majority of us probably. We're in urban pollution, air pollution. It's just, it exists. Even if we've cleaned up some, it exists. And we're eating oftentimes food that we haven't prepared ourselves from our own backyard and our own garden. And so all of those changes that are common here, I think, have contributed to a degradation that is not just about a clinical diagnosis, but when you start to look at even, I mean, there are receptors that actually, I think some of the taste receptors or taste receptor bacteria, or maybe it's the olfactory receptor. Olfactory receptor affects cardiovascular status as well. How's that work? So these microbes are doing things. We're still learning about the connectivity between them. But I wanted to point out, it is actually very difficult for us to socialize in a way and to experience nature as fully as I would contend our grandparents used to do, particularly here. Again, the U.S. is mainly what I'm referring to. So thank you for letting me clarify that. Clearly, other countries have their own issues with status of the microbiome, and some of it's not countrywide. It's, you know, it depends on where you are and what you're doing. But I do think that it's eroded things that we may take for granted, we may not realize. Are we smelling and sensing things like we used to? Are we tasting like our grandparents did? And that's the zest of life.
It's very true. So we're going to get a couple questions in. One right next to you, right there. One in the back with your hand high there. We'll take these two, and then we'll see if we can get two more after that.
Thank you for this fascinating presentation. I'm struggling to reconcile the idea of taking a single microbiome or single microbe and spraying it on your face with the idea of the terroir, right? And how do we build a world where we're actually generating these microbes that we need to live a healthy life, right? In that terroir kind of way, as opposed to taking one and importing it. Thank you.
And then this other question here. Stand up, please.
Yes, hello. First off, I will try to be brief, but thank you so much to the organizers of this conference for making it freely available to anybody who found out about it and were able to register for it and get ourselves here. So thank you. And I want to pick up on a theme, I think that's probably perhaps the purpose of why you organized this, which was to reach out to young people who are at the outset of their careers and looking to potentially enter this field. Well, I come to this as somebody who's at a turning point in my career and also looking to potentially enter this field with a general biomedical kind of background as a seasoned professional. But I guess what I'm kind of interested in bringing up and maybe broadening that theme and also seeking some concrete guidance from those in the room who perhaps are already part of the field to those of us looking to enter the field. What do you see as major opportunities and gaps? What thoughts might you have on areas of skill development, or are there urgent needs for people with particular skills? I guess it's kind of an open question, but it's really seeking some kind of concrete career guidance, not just for those at the beginning of their careers, but perhaps at the turning points of our careers as well.
So great question. And for time reasons, I'm going to ask Larry to answer the first question about the one microbiome, and then let Laura and Rodney take a stab at the second question.
Yeah, so the example with AOBs, Nitrosomonas eutropha specifically, it was kind of a softball. The fact that it was one. Bacteria, by the way, never exist in monoculture in nature, ever. The fact that we had this one, it took us from a place where we never thought about such a type of intervention to one where we had an example of it worked. And by the way, once that was there, it only persisted, at least in our study, it had a half-life of about a week to a week and a half before you'd have to put it back. So for most of these, if they're not in our terroir, they will probably do best as a continual application, whether it's of live bacteria or of the genetic or metabolic machinery. The flip side of your question is we live in built environments full of xenobiotic chemicals. Xenobiotics are chemicals that are not natively part of our physiology, by the way. If someone says their product doesn't have chemicals in it, they better be selling software. So when we live in this environment, we will adapt to this. If I swab the walls, I'd find skin bacteria. But we should start thinking about whether or not, since our carpet is going to have bacteria live on it, maybe we want to build a healthy microbiome on there instead of try to apply chemicals to kill things and feed our carpets.
I love this. It's like the Internet of Things is now going to become the microbiome of things. Do either of you want to respond to the question about where one goes in the field and how one enters this field?
Well, I would just say that most of my career was working in immunology and toxicology, particularly protecting the immune system of children. And I only entered the microbiome field by having a dream in 2012, writing up the concept of the dream, and that led to so many other papers and invitations and a documentary movie and a book offer with no proposal and things that you wouldn't even, how could you possibly plan your career that way? So don't limit yourself. You've got a different perspective, and that's really going to be important. You have people that are slugging away doing brilliant research and have been doing it since the beginning in the microbiome, but they need to encounter people with different perspectives because that's going to drive applications we may never have dreamed of.
And cold call them. Find people you admire, call them up, go meet with them. You'll be amazed. You bring whatever your career has been, you bring value to the table.
Okay, we're going to have two last questions. One Martha and one right up here, this gentleman in the second row.
Mine's not really a question, it's a response to you because I'm somebody who came from out of left field into this field. So I would say if you have passion for it, and he's saying cold call, I mean, some of the people who have come to work for me are people who showed a tremendous passion and they didn't come particularly from the field, but they came in and they worked with me and learned the skills because they had the passion. And what I would say is in the field of microbiology, it is very collegial. And if you're interested in it and you want to meet the scientists without actually having to meet them, I mean, to go places, they are on Twitter. If you look up microbiome and microbiology scientists on Twitter, they share all the papers, they share information, they pick the details apart, and they will talk to you as a citizen scientist, as somebody from another field. And I think that's incredibly important because they've created an ecosystem that is open to this other thinking. So I would just suggest that you try to start there.
Great. Last question.
Hi. So I know Dave. Sorry.
Stand up.
Oh,
Do you mind?
yeah, no problem. So I know David's situation was different because he had a mother who was indigenous and a father who was from Western kind of culture. But if one of us who were kind of engulfed in Western civilization all of our lives, and we would up and go to one of those places, how would you think that affects not only the individual condition, but also the social condition of where we are right now in the microbiome?
Yeah, it's a very tough thing because when we show up there, historically, just bad things have happened. And we bring diseases and other things that they don't have. On the other hand, if we don't do anything, and the general consensus among people who really understand the problem is that they're not going to survive. And so what I would say is we need to be creative. We need to be, what the Buddhists say, be helpful, be truthful, and be kind. And somehow the best thing that we can try to do is to help these people survive. And maybe that intervention is, start a conversation about why, for purely selfish reasons, which seems to appeal to people most, their survival is important to our survival. But whether we actually go into their land, I have made a decision not to ever do that, even though David wants me to. I don't think we belong there. It's just that simple for me. But look, you guys are smarter than we are. You got lots of time on us. Find a better way. There's got to be a better way.
So first of all, let me ask you to thank our panelists up here for it was a great presentation. So we're going to jump right into our next panel, which is on telling the truth in science.